So welcome back everybody. So we've already talked now about disaccharides and the linkages that you can have between sugars. And then we started talking about polysaccharides. So let's continue the conversation about polysaccharides. Remember the last one we talked about was starch and how it has amylose and amylopectin and how they're different based on the different type of linkages they can have, right? So far we've seen a 1,4 alpha and also a 1,6 alpha. So let's continue on with that conversation. If that doesn't sound familiar, make sure that you go back and watch that previous video so that way this will all make sense, all right? But let's move on to glycogen. All right, so now let's talk about glycogen. What's cool about glycogen is it has the exact same things as amylopectin. It's got alpha glucose in 1,4 alpha linkages. It's got alpha glucose in 1,6 alpha linkages. So it's the same thing. If you look here on the bottom, it'd be the same setup as amylopectin, right? That looks the same. So again, it's making the alpha helix by making the 1,4s, and then it's going to start making branches by making the 1,6s. What's different about it is that it actually has more branching than the amylopectin does. So it has it one every 10 sugars is what they say. So that's pretty frequent. So as you can see over here, it's branching out in kind of a cool pattern. And there's also the branches are shorter. They're not as long. So it's not trying to be like long and spindly. It's trying to be short and compact. And that's good because it's more compact. So think about, if you're thinking about humans versus plants, who would need the most energy? It's probably going to be humans and animals, right? So why would they need more energy than a plant? What do they do that plants don't really have to do? They have to move, right? They have to get moving. So a plant doesn't really have to move around. It can gently move like to face the sun or things like that. When it grows, it moves a little bit, but generally it's not moving around. Whereas humans and animals need to constantly be moving to keep up their metabolism, to catch prey, all these different reasons. So they need something that's more energy dense. And so more compact equals more energy, and that's why you need that for glycogen. So just imagine more branches makes it more compact, more energy. Animals need energy. Good? And that's kind of an easy way to remember it. One more thing we want to talk about is the purpose of glycogen. So as we said, it's to store energy also, just like plants need, but it's just a little bit different information. So how humans and animals store sugar is the purpose of glycogen. And then generally what it's doing is it's storing sugar from after you eat, and then it's putting most of it in the liver because the liver's there to regulate your blood sugar level, right? That's kind of its purpose. That's where the insulin is. That's where all that stuff is happening. So that's what's happening in the liver. It also takes a small amount of the glycogen and stores it in your skeletal muscles. And this is for movement. So like if you suddenly need to move, it's already got some energy ready and waiting to do that job, right? The other thing we want to point out is that this is for short-term storage. It's not like fats where you're going to have it for weeks and weeks in case you need it later. It's really for short-term, like during the day. So that's about 8 to 12 hours. Just think about like in between meals, right? Like you really shouldn't go more than 12 hours without eating. But generally you can if you're sleeping or whatever. And so generally that's what it's doing is whenever you aren't eating, it's converting it to glycogen to save it and then releasing it slowly over time. And then you run out eventually and you need to eat again. So it's a normal process. So we have one more type of polysaccharide to talk about, and that's cellulose. So cellulose is one you probably heard a lot about. Cellulose is found in plants as well, but it's not used for the same purpose. Before we talk about the properties of cellulose, let's talk about what makes it different based on the structure. So if you look at the bottom picture here, I've got a picture of the linkages. What type of linkages are these? Right, they're 1,4 beta. They're still 1,4 because this is carbon 1 and this is carbon 4, right? But then they're also, this one's going up on the one carbon. Remember, it's the one carbon that determines if it's alpha or beta. It has to be the place, the key carbon, right? The place where there's two oxygens attached. So it's the key carbon that gets to decide. So because it's the one carbon, it's going up, that's going to be beta. So 1,4 beta linkages, all right? So then if we look at that, what that structure is going to look like at the top is it's going to be long chains, just like the alpha helix ones, right? They're long chains, but because they're going up and not up and down, then that means that they're actually going to be pretty flat. And because of that flatness, they don't start curling. And then you get this hydrogen bonding across the different lines, right? So that's what makes what's called a beta sheet, or you can call them a cellulose sheet. So that's going to have different properties, right? Because it's going to be flat and compacted down instead of being like spindly and long and twisty, okay? So then what this means for the plant is that this is actually going to be a structural element rather than a way to store sugars. Because it's difficult for even the plant itself to break it down, it can't easily get those glucoses off to use it for energy. So instead, it's going to use it to be strong and structural. So that's what's going to hold up your stem to your plant, for example. It's going to give it that thickness that we're talking about, right? And so, what this means is that larger animals and humans, especially, are not able to break down 1,4-beta linkages. We're just not able to do that. That's not part of something that we have. We don't have an enzyme for it. We can't do it. And so, instead, even herbivores don't have it. They have to use a bacteria. So there's a special bacteria that has an enzyme that breaks down the beta linkages. And then if you're an herbivore, you would have a lot of that bacteria in your stomach. And then in some cases, you might even have more than one stomach, like the cow that has 4 stomachs, right? It needs all that stuff to get enough energy out of the cellulose to actually live and support its body weight. So that's kind of a cool fact, is that we're never going to be able to break down cellulose unless you have that bacteria. So that's why we just don't have that, and why we don't get a lot of our energy from, say, eating a salad, which is why salads end up being diet food, right? Because you eat them, but you don't really get a lot of energy out of them. And it sort of also cleans out your system. It's got fiber in it. So that's another thing that cleans out your stomach. So, more fun facts about your food and this chemistry. So let's pause and review and see if we can answer a question about some of this material that we've covered so far. So the question is, which of the following does not have an alpha helix in it? So pause the video and think about the answer to that. All right, so what do you think? Which one is it? Right, it's going to be cellulose, right? It doesn't have an alpha helix because it doesn't have an alpha linkage, right? Alpha linkage, alpha helix, okay? So it's got to be cellulose. Amylose and amylopectin both have 1,4 alphas. So does glycogen. They all have 1,4 alphas. And then amylopectin and glycogen also have the branching, which is the 1,6 alpha. Less to do with the alpha helix, but still relevant, right? So it's just going to be cellulose. So one more polysaccharide I want to mention. This is not going to be on the test, so it's just for fun, but it is related. So there's one more polysaccharide that you might hear about, which is called chitin. Chitin is found in the exoskeletons of bugs and also the cell walls of fungi, right? So if we're looking at chitin, let's look at the structure. As you can see, it's a little bit different, right? So what's totally different about these polysaccharides? They're not normal sugars, right? So if you look at the bottom, instead of having an OH where I should on carbon 2, I've got an amide there. So that's a little bit different, right? That's why I'm not really including it in this section. It's also not as relevant to human biology, but it's interesting. So basically it's got this other group at the bottom, so that's going to totally change how it can pack and how the structures can form and things like that. If we're looking at this, what type of linkage is it going to form? So we know that it's between this carbon and this carbon, so that's going to be what numbers? 1, 4, right? And then is it going to be alpha or beta? So I know it's drawn a little bit weird, but if you look from carbon 1, is it going up or down? It's going up, right? So then that's going to be beta, right? So beta goes up and then alpha goes down. So this is beta. So just like we saw with the cellulose, if you have a beta linkage, that's going to be more about structure, right? So these are, it makes sense, it's going to be about structure. If it's an exoskeleton, it's going to be super tough. So these are used for exoskeletons, so kind of cool. Generally, you're not going to be eating these a lot unless you eat a lot of mushrooms. Or also if, fun fact, there was a while back at Starbucks, they had this drink. It was like the Frappuccino, the strawberry Frappuccinos, and they used to be really pink. The pink in there was actually made from some sort of shellfish or something that had an exoskeleton. So you might have been eating chitin if you ever had that, which I've had lots of. It didn't hurt me. It was fine. But so generally, you're not going to be eating a lot of chitin in your diet. So you don't need to break it down. It would just go through you like fiber would, right? So it's kind of a similar idea. All right, so then that's just chitin. I just wanted you to at least recognize it. If you've ever seen it in the future, if you hear about it, you'll know a little bit more about it. And so with that, we're done talking about our polymers, right? Our polysaccharides. So just a reminder, these are the 4 we need to know: amylose, amylopectin, glycogen, cellulose. And then what you need to know is the sugars, the linkages, the tertiary structures, and then the properties and functions of the starch, glycogen, and cellulose, okay? And when let's look at the pictures, these are what they look like. You should be able to, from the information you have, be able to recognize them. So like if I just showed you a picture, you should recognize what those look like. That's another cool thing that you can do based on the information that we now know. So just make sure you can do that. And then the last thing that I would do is I recommend making a table just to organize the information. So basically I would arrange it like this. I've got my disaccharides on top, my polysaccharides on the bottom, right? And then I'm going to talk about which ones that they're made of, so the sugars that they're made of, so put that in there, and then also the types of linkages, so just a way to keep things straight. It doesn't have the properties, so you might want to either put that, say, on the previous picture that we just showed, or you might want to put that in a separate spot, or you can make a little spot. Just know amylose and amylopectin are used to make both glycogen and starch, and those will be the properties you'd be looking at, okay? So hopefully this was helpful. Make sure that you go over this material before the exam, and it's very conceptual, so I know if you're a sort of more of a bio type person, hopefully that'll be a little bit easier to make some note cards or a table or whatever you want to do for that, all right? So that's going to be the end of our video, and make sure you go and try out the activities that are posted, and let me know if you have any questions. Thanks everyone. Bye.